Thank you for your kind introduction, Claudia. Good afternoon, everyone. My name is Atsushi Sekiya from Funakoshi company in Japan. I'm a product scientist in Funakoshi for commercializing and developing research agents, especially innovative Japanese fluorescent dyes. This time, we gratefully appreciate uh, Euro-BioImaging Consortium giving us a chance to talk in the tech exchange webinar. I'm very glad to have my presentation here. Today's my topic is imaging method for sensing membrane microenvironment in living cells. Also, today's excellent webinar from Dr. Charaps, and I believe my presentation will be well fit on interest of today's audiences. This is our company profile. Funakoshi is a Tokyo-based Tokyo-based Japanese company established in 1923, and Funakoshi has 100 years history. Our business model is distributor of licensed reagents from worldwide into Japan, but recent 10 years, Funakoshi is focusing on commercializing and supplying innovative research reagents, especially fluorescent dye originally developed by Japanese academic researchers. Here shows some example of our innovative fluorescent dyes. Among dyes especially repeat research tools were growing in Japan, in US and world. So for example, ultra photostable and ultra ultra photostable and ultra sensitive repeat droplet dye for long-term lifestyle imaging, and color changeable fatty acid metabolism tracer. And this one is a fluorescent probe for measuring cellular activity for fatty acid oxidation and so on. And we also have a unique chemogenetic platform controlling protein localization between plasma membrane and cytosol by a small compound. So today, among our popular and unique lipid metabolism tools, I'd like to show lipid order for sensing microenvironment of cellular membranes. This dye changed its fluorescent color from green to red, depending on its local environment, like this picture. So here is the content of my presentation. In this presentation, I am to provide a comprehensive description of the scientific background and conventional method or principle of this dye and application note for sensing membrane microenvironment. And in the later presentation, I'd like to also talk a little about 3D imaging of tissue structure as an advanced application of this technology. So at first, I would like to show a scientific background of membrane microenvironment. As you well know, cellular membrane, including plasma membrane and end membranes of organella, are made up by lipid bilayer. This lipid bilayer consists of a huge variety of lipid species, such as phospholipid, glycolipid, and sphingolipid, and so on. Among them, phospholipid is the most major category of lipid and plays a central role in the creating cellular membranes. So next slide shows a breakdown of phospholipid variation. Phospholipids have usually two variable modules. One is hydrophilic head group, shown blue, and Another one is hydrophobic fatty acid tail yellow. So because of the diversity of fatty acids, there are theoretically many possible combinations of phospholipid. So fatty acid can be roughly classified into the two categories. One is a saturated fatty acid without any double bonds, and another is the unsaturated fatty acid with one or more double bonds in the long carbon chain. So diverse combination of phospholipid is considered to create several structures. So from here, I'd like to focus on the local structure of lipid bilayer. This picture shows two types of phospholipid. Green lipid shows a phospholipid composed exclusively of saturated fatty acid. So these types of fatty acid, these types of lipid have a linear and closed structure. And on the other hand, red lipid shows a phospholipid contained unsaturated fatty acid, like this picture. And these types of lipid have bent and open structure. So what do you think happens when two types of lipid are mixed together? So in the simplest model, these two types of phospholipid do not homogenize with each other and are known to separate into two phases. And this is called phase separation. In this model, lipid bilayer containing green lipid, so saturated phospholipid are generally known for the dense structure, thickness, and low fluidity. On the other hand, the red lipid contained unsaturated lipids are known to have a sparse structures and thin and high fluidity. So historically, these green lipid bilayer called liquid ordered phase, so called LO, and this red lipid bilayer called disordered phase, LD. And this biophysical parameter is often called lipid order, meaning the order of lipid bilayer. So lipid order is one of the important characters to discuss the lipid raft theory, and lipid order also well correlated with membrane characters such as membrane curvature, fluidity, and thickness, and so on. So this model is very simple because there are only two typical lipids, including saturated and unsaturated lipid. However, in the actual cell, there are a huge variety of lipids, so many more complex events in the cell. So here is a model of a complexation of heterogeneous membrane structure. So in my presentation, I'd like to call this difference of membrane composition to membrane microenvironment. In many cases, several local structures have different membrane microenvironments, and understanding of membrane microenvironment is a very important point for barrier cell functions. So if we can sense membrane microenvironment easily, we may access barrier cell function by imaging technique. So to sense membrane microenvironment, how should we do? I move forward to next section. So next slide is one of the answer for sensing membrane microenvironment. A correlation between membrane lipid composition and water molecular contents have been proposed as one solution for sensing membrane microenvironment. So as you know, water molecule, so H2O, is one of the highest polar molecules. The water molecule play an important role to determine the physical character of lipid bilayer. In the LO phase, water molecules are excluded because of its higher hydrophobicity and tightly packed structure. Just opposite, LD phase shows a sparse space by its open structure, and water molecules like incorporated in the lipid bilayer. So the middle layer sought to gradually increase what increase in water content from LO to LD. So LD phase reach water molecule and LO phase very little water molecule. So membrane microenvironment can be monitored via quantitative observation of local polarity derived from water molecule in the lipid bilayer. The solvent chromic dye are historically utilized to quantitatively monitor molecular polarity. So typical fluorescent dye, Rauden and NEPPDHQ often utilized to sense the membrane microenvironment because these solvent chromic dye can be accumulated in the lipid bilayer. However, Rauden required UV excitation and not suitable for lab cell experiment. And there are some disadvantages of this conventional dye into the lab cell imaging. So our repeat order is a novel solvent chromic dye, originally reported as a PK probe in 2020. This dye emits visible light fluorescence from green to red. So from here, I would like to introduce sensing principle of repeat order. I think this picture promotes your understanding of solvent chromic character of repeat order. So when repeat order solvarized into various organic solvent, repeat order show different fluorescence color depending on its solvent polarities. So under low polaric solvent, it emits cyan to green, and the higher polaric solvent shows the orange to red. However, absorbance spectrum is not changed in the difference of solvent, so we can excite the repeat order by a single laser, for example, 405 nanometer laser. And another important character of repeat order is accumulated in the lipid bilayer. So combining these two features, repeat order can sense chemical polarity on the lipid bilayer like this picture. Actually, using an in vitro liposome experiment revealed LO model liposome show green emission and LD model liposome show red. So furthermore, a liposome with intermediate properties shows result in between LO and LD. So this is experimentally measured fluorescent ratio red per green, and this ratio value is well correlated with model LO and LD. So next, I'd like to introduce two imaging results of lab cell imaging. Here is an example of lab cell imaging using COS7 cell. COS7 cells were stained by 100 nanomolar repeat order, and fluorescent imaging data were observed by confocal microscopy excited by 405 nanometer laser, and detection window are green and red. So merged image further analyzed the ratiometric method by ImageJ software. So this picture is an example of ratiometric analysis of repeat order. To see more in detail, next, this slide shows analytical panel for further ratiometric analysis. So this panel shows individual ratiometric layers separately. So each layer visualizes unique organelle structures. The most lower value, so most nearly L phase shows clearly shows a plasma membrane structure, and a higher ratio value, and this one shows the typical mesh structure of ER, and the highest ratio value shows mitochondria-like structure. So this result indicates the Lipiodda is distributed to almost all organella and can sense local membrane microenvironments. This is another example of Lipiodda staining for primary cultured neuron, cell bodies, dendrites, and axonal terminal. So likely to cause 7 cell, plasma membrane is basically L phase, and the end membrane are LD phase. So I hope Lipiodda will be promoting tool for promising tool for membrane research by to easily catch the structure or local structure of cellular membranes. So by the way, many researchers ask us whether Lipiodda is compatible with tissue local structure in 3D imaging. Actually, answer is yes, but Lipiodda is not the best for the tissue application. So in this time, I also introduce another dye, Hisprite. This is a sister compound of Lipiodda, but very specific for the tissue imaging. So the final section, I'd like to introduce, briefly introduce tissue structure imaging by Hisprite. So like Lipiodda, Hisprite has also solvatochromic character and show more broad spectrum from green to far red. So you can easily see the difference of Lipiodda and Hisprite. And furthermore, Hisprite is more hydrophobic dye than Lipiodda and highly fit for the penetrating thick tissue blocks. So combining with tissue clearing technique and two-photon microscopy, Hisprite clearly visualizes tissue 3D structure by only single dye. So this is the actual fluorescent imaging data of paraformulated fixed human normal skin cell tissue combined with lucid tissue clearing method. So cyan indicates the signal from second harmonic generation derived from collagen fiber. So please note this is not from Hisprite, but right 3 images were obtained by two-photon microscopy excited by 960 nanometer two-photon laser and detected 3 ranges. So using these 4 images, the merging image were created here. And this is an enlarged image. We can see the epidermis, dermis, sweet gland, nerve fiber structure, and with extracellular matrix. After Z-stacking imaging, the 3D image were reconstituted here. So 3D image give us spatial information of tissue local structure, like this picture. So in this experiment, we use only Hisprite, no additional dyes, but we can see beautiful and clear structure of human skin by deep imaging. So this is a conclusion. In this time, I showed two types of solvatochromic dyes. One is Lipiodda for sensing cellular membrane microenvironment, and another is Hisprite for visualizing the tissue 3D structure. So we're a Japanese company, but both dyes can be available in European countries via our local distributor. So if you have any if you have any interest, please check our website or send me email. So here I'll put the discussion. So if you have any question, I'm glad to discuss now. So thank you very much for kind attention.